Well, hello, Chemistry 130L. Now we're finally going to do some wet chemistry. The first experiment that we'll be doing is understanding the fundamentals of chromatography, doing some work with some food coloring and some Kool-Aid. Okay, so before you get started, you'll want to make sure that you have your lab protocol. On page 4 and page 5, it will list out all the supplies that you need. So one supply you'll need will be the little baggie marked Fundamentals of Chromatography. Inside that bag, you'll find 3 packets of Kool-Aid, 6 pipettes that have a little extra tip on them, and 6 sheets of chromatography paper. Now, I've already opened mine up because I went ahead and got some of the experiments started so that as I'm explaining things to you, the experiment has time to run because it takes about 15 minutes for each chromatography experiment to produce its chromatogram. And so I didn't want to set it up and then sit here for 15 minutes doing nothing. So I already have part of the experiment going. From your equipment kit, you need all sorts of things. You need your test tube rack, your test tubes, one of your pipettes, your balance, graduated cylinders, ruler, beaker, the small plastic cups, weigh boats, your plastic spoon, your packet of food coloring from your chemical kit number 1, your straight pins from your chemical kit number 1, and then a whole bunch of stuff from around your house. You need some purified water, table salt, a pencil, this is really important, scissors, paper towels, aluminum foil or a plastic bag, white paper, tape, toothpicks, and then for activity 3, you'll need something else to test. So M&Ms, Skittles, an ink pen, just something that you can use for activity 3. The other things you'll want, it's not listed, but you'll want your grease pencils because you'll need to do some labeling. And you'll also want an ink pen because you do need to record your data in ink. There are some things that you'll need to do in pencil, which is why you need a pencil, but you absolutely will need a pen to record some things in ink. Okay. The other thing you'll want is to print out your report sheet with your data tables so that you can record things as you go. All right, so to start with on this experiment, we're trying to understand what's inside these Kool-Aid packets. And then in activity 3, what's inside the other item you choose. And so what we're going to be doing is we're going to be doing some color chromatography with our food coloring. So we're going to take our red, our yellow, and our blue food coloring and produce known chromatographs of those pure colors. And then what we'll do is we will test our Kool-Aid, and then we'll test our chosen item because we'll need to calculate something called our retention factor. And our retention factor is R sub F. Okay, and what we're looking at with our retention factor is the distance our analyte or our solute travels divided by the distance that the solvent travels. Okay, so distance distance analyte travels over distance solvent travels. Okay, because what ends up happening is you'll have your strip that you blot on. So you'll have a line drawn and you'll have another line drawn. Okay, this bottom line will show you where to spot. This top line will help you figure out where to stick your straight pin through to have it suspended in your solvent solution. So let's say we're doing our red. Okay, so our red, we're going to dot here to begin with. Okay, and then we're going to place it in our solvent. And over time, what'll happen is they'll travel up our strip. So our solvent will fall somewhere down here below our red line because we don't want the solvent to immerse that dot. We want the solvent to hit our chromatography paper and have that be what causes the dot to travel up. And so throughout the experiment, okay, eventually that dot will travel, okay? And so will our solvent front, okay? And so what we'll do is we'll measure the distance for our dot and we'll measure the distance for our solvent. Okay, so let's say that this moves 4 millimeters and this moves 10 millimeters. So my retention factor here, I'm going to take that 4 millimeters for my analyte divided by my 10 millimeters for my solvent, and I would get a retention factor of 0.4 for that particular analyte. All right, so that's what we're trying to do with this experiment. Now, for this experiment, we're actually going to have you mark the midline, because the way these colors go up the chromatography strips is they'll eventually start spreading out a lot. And so the actual good movement of your dot is going to be kind of in the center of where that analyte travels. All right. Okay, so we have to do some prep work first. All right. So before we start actually experimenting, we need to make sure we have our PPE on. So I'm going to take my lab coat and I'm going to go ahead and fasten it. Okay. So my lab coat buttons, my lab coat snaps, if yours buttons, that's perfectly fine as long as it fastens. Remember, you do need a cloth lab coat, not the apron that comes with your lab kit. So I'll snap that up. Okay, I need my safety goggles, so I'm going to put those on. Okay. And then I need a pair of gloves, so I'll go ahead and get a pair of gloves out. Here's the open package. All right, so there's my gloves. These will be really nice with this experiment because we are working with food coloring. And so if you accidentally get some on you, it turns your skin colors for a while. But if you have gloves on, it'll prevent that. All right. So, and I like to make sure that my shirt sleeve goes over my glove. That way everything's nice and tucked in. Okay. All right, so the first thing I want to go ahead and start prepping is my Kool-Aid. Now, I've already prepped 2 of them because, again, I wanted to get stuff running so that I can show you a little bit about how to find that center of your solute front when we are done here. So I labeled my cups. So I took my grease pencil and labeled my small cups S-O-N-G for strawberry, orange, and grape. So the one that I haven't done yet is strawberry. So what I have found with these that's easiest is you just cut off a corner like that. It makes it easier to pour it into the weigh boat. So what I'm going to do is I'm going to take a weigh boat. I'm going to turn on my balance. Okay. I'm going to put the weigh boat on. Now, when I put the weigh boat on my balance, I end up getting a reading. Now I can add my Kool-Aid to this and I can try to subtract it in my head, or if I hit the tare button, now my balance reads 0. So it's going to make it easier to weigh out my Kool-Aid. Okay. And then you open up that little slit that you made. Okay. And now it tells us that we need 2 grams. So I'm going to carefully shake out Kool-Aid until I reach that 2 gram point. Now for this experiment, if you get a slight bit more or a slight bit less than the 2 grams, it's not the end of the world. So like my balance, if it reads like 2.03, that's fine. I don't need to get it exactly 2.00. All right, I'm gonna pick up my weigh boat. I'm gonna bend it in half a little bit. I'm gonna pour that into my little cup. Okay, and then I'm gonna put it over here with my used weigh boats so that I can wash those when I am all done. Now what it tells me is I need 5 mils of water. Now here's my jug of water. I find it really difficult to pour water like this into something small. So I've actually put my water into a plastic cup, and I'm going to use my graduated cylinder to get 5 mils. Okay. Now, if I want to make sure I'm getting a good 5 mils, I'll lean down and I'll pour in this way. And I'll read my meniscus at eye level. Hopefully you remember that from Chem 100. Okay, and then I'm going to go ahead and pour that in. Okay, and then I can swirl it to mix it. Or if I've got clumps of stuff, I can take my plastic spoon and I can mix it that way. Just make sure that you rinse your plastic spoon in between each Kool-Aid. All right, so I'm going to do that for all 3 of my Kool-Aids. Okay. All right. The next thing is in your beaker. Okay, you'll want to do your salt. So you'll use another weigh boat. Turn your balance on. Hit the tare button. I've put some salt in another container here. Now, 0.1 grams of salt isn't very much, so be careful when you are dispensing your salt. You'll want to then pour that into your beaker and use your 50 ml graduated cylinder twice in order to get your 100 mils of water that you will add to make your 0.1% solution of sodium chloride. Now, again, I already made mine because I wanted to get my experiment going. All right. So I'm done weighing stuff out. So I'm going to put my balance over here so it's out of my way. Okay, now the next thing you want to do is you are going to prepare your chromatography strips. We gave you 6 strips in the kit. You want 12. So what you're going to do is you're going to be cutting these in half. Now, you also need to mark some other lines on them. What I found is when it tells you to mark lines at 1.5 and 9.4 centimeters, I found it easier to mark those lines before I cut the strip in half. So I'm going to take my ruler, and on the metric side, I'm going to use a pencil. It's very important on your chromatography strips that you use pencil only, because you don't want any ink in the pen to travel up. So you just want to use pencil. So I'm going to mark my 1.5. I'm going to mark my 9.4, flip my ruler around, and I'm going to draw lines. Okay. All right, and then I also found it easier to draw a line to split the strip in half before I cut. So I drew my line, and now I'm going to cut my strip in half. Okay. All right, so I cut my strip in half. And then the instructions tell you to also cut a little V shape on the bottom. Now, the reason why we have you do this is just so that you don't confuse the top from the bottom, because that top line that you drew is your marker line to know where to put the pin through so that your strip sticks out of the solution enough so that, again, your solvent is hitting down here and not where your dots are. So the whole point of those little arrows that or the little V shape is just to make sure that your strip sticks out enough. Okay, and then the next prep you need to do is with the pipettes. Okay, so for the pipettes, you don't need to actually squeeze the 6 pipettes that came in your chromatography kit, the ones that have the little extra tiny tip on the end of them. Okay, so for those, I'm going to use a Sharpie, and I'm going to mark 5 centimeters from the tip, just with a little dot there, so that I know where to cut my pipette. Okay, start that, because all I need is this little piece. Because when you do chromatography, we just put little bits on our whatever we're using for our chromatography, in this case, the chromatography paper. And so we use capillary action up through tiny little pores like this in order to just pull up a tiny bit of our analyte and then dot that onto our chromatography medium. Okay, all right, so that part is done. All right, now I'm going to do my known dye. So I already have my red and my yellow running, so I'm going to do my blue. What we are having you do is, because if you try to take this and put a dot directly onto your chromatography paper, it's just going to oversaturate it. So what you want to do is on that foil or on your plastic bag, just put a couple of drops of each color so that you have some sitting there. Okay. All right, and then you'll take one of your chromatography strips. Now, making sure that you know which one is which, at the top of it, okay, above that top line, write what you're dotting. So this one, I'm dotting the blue food coloring. So I'm going to put that there. And then to dot it, what I do is I take my pipette and I'm going to put it in there and just let a little bit run up it. So you probably can't see this on the video, but there's a teeny tiny bit on my tip, and I'm going to then touch it to the bottom where that line is. So again, that line that you drew at 1.5 centimeters, that's where you want to put your dot. So here, let me come closer to the video. I'll show you what it looks like when you do this. Okay, so you can see there's my blue dot. And then there you can see that tiny bit of food coloring that's in my pipette tip. And so again, it's just capillary action, a little bit travels up, and then when you put it on the paper, that same thing, the paper attracts the dye better than the pipette tip does, and so it flows out of the pipette tip onto the paper. Okay. And so you want to do that a couple times to make sure you get a good dot. And then you'll want to let it dry for a minute before you put it into your solution. Okay, all right. And so once you've let it dry for a minute, then you'll take one of your straight pens and you will stick it through that top line in the center there, like this, so that it's hanging. So that way you can do your experiment. So what you're going to do is in each of your test tubes, you're going to take some of your 1% sodium chloride solution and you're going to drop in 20 drops. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. Okay, and what that looks like is just a little bit at the bottom here. 20 drops is approximately a milliliter. Okay, all right, so now I can hang that strip in there. Then again, you want to let that go for about 15 minutes. Okay, so you're going to let each of those go. And after your 15 minutes, you're going to go ahead and take them out. Okay, so here's one of them. Okay, so now what I need to do, okay, is I'm going to go ahead and put this on a paper towel. I'm going to remove my pen. And so what I want to do is I want to look at this and I need to see where my solvent front. So my solvent front, you'll be able to see, even though it's clear, because it will look wet. So let me again bring this up to the camera better. Oh, and my solvent front went basically to that line. Okay, so you can see where you can tell that it's wet on top of that color, and then you can see it's still dry on top there. So it's easy to tell the difference between the wet and the dry. And so I want to mark where that solvent front stopped. Okay. And then in the middle of where my color went, I'm going to make a mark. Okay. And so then I'm going to do that for each of my 3 food colorings. And then I'm going to take my ruler and in millimeters, I'm going to measure from my line to that middle line that I drew. Okay, and that will be the distance my analyte traveled. And then I'll measure from my bottom line to where my solvent front stopped, and that will be the distance my solvent traveled. And that's how I will calculate my retention factor. And I'll do that for each of the 3 food colorings. All right, and then you're basically going to do the same thing for your Kool-Aid. So if we go to activity 2, okay, so again, we're going to have our strips. Okay, I have a couple of them going already. So the one that I haven't done yet is that strawberry. So on the top of my strip, I'm going to label it strawberry. Okay, and then this time, okay, instead of a little dot on my foil, I can go ahead and put it in here. And again, a little bit will get into my tip, and I'll be able to dot that on there. Okay, I'll do that a couple of times. Okay. All right, and then I can put my straight pen through there. All right, and put that in. So what you should do is you should prepare your chromatography paper for each of the 3 known dyes, and then put all 3 in at the same time. That way your 15 minutes all goes at the same time. Take them out, and then you should prepare your 3 strips for your Kool-Aid, put all 3 in at the same time, start your 15 minutes, and then you can take all 3 out at the same time. So when you're all done, you're going to compare your chromatograms of your Kool-Aid to that of your 3 known dyes, your red, your yellow, and your blue, to try to analyze what might be in the Kool-Aid. Now, this is a good experiment to do, especially if you're like me and you're allergic to, certain food colorings. I'm actually allergic to that red number 40 dye. I can't eat it. It doesn't work for me. And so if I had, a sample of something, some food item that didn't have the food colorings listed as an ingredient, I could do an experiment just like this to figure out what food coloring existed in that particular food item to see if it was safe for me to eat or not. All right, so I'll do all that. And then activity 3, all right, you're going to take what you have learned from activities 1 and 2, and you're going to go pick another food item or a marker or something around your house that has colored dye in it that you can dot onto your chromatography paper and then run this experiment. So you need 6 strips of chromatography paper in order to do the main experiment. Because you had 6, you cut them in half, you did it with 12. So you have 6 more sheets of chromatography paper to do activity number 3 with. If something happens to your chromatography paper and you run out for some reason, or you have to rerun something, or you accidentally spill something, you can use coffee filter. The coffee filters, those will work the same way. Okay, so on my data tables here, in ink is where I'll record my data table. So when I did my red number 40, I looked at this. Okay, what is the color of my band? The color of my band, I'm going to write red, and I'm going to write that in ink because we always record data in ink. Okay, distance the band traveled. So again, that's where I'm going to take my ruler, and I'm going to measure up to that bottom line. So on mine, middle of mine was, looks like 4.5. Okay, and then I can read to 2 decimal places. So I'm going to write 4.50 because this fell right on that line. Because remember this from Chem 100. We can take our equipment and we can read 1 past whatever our marks are. And that last digit is our uncertain digit. So my eye tells me that that fell right on the 4.5 line. So I'm writing 4.50. Maybe I'm a little bit off. It could be 4.49. It could be 4.51. I'm estimating that visual, cue there that says this was right at 4.50. Okay. And then I don't need to put units because my data table at the top of the column already has millimeters. And I'm going to do the same for my solvent. My solvent traveled just past that top line. And again, I'm measuring from that bottom line, not the bottom of my paper, but that bottom line that I drew, because that's where I dotted my analyte. And I'm going to look here, and this looks like it's at 8.11. Okay, and so then for mine, okay, for that retention factor, I'm going to take my 4.50 divided by that 8.11 millimeters, and that will give me my retention factor for my red dye. Okay, now when you do this, pay close attention to what you see, because I have space on here for observations. As you're doing your experiment, write any observations. If something happens to your experiment, write your observations. Okay, it's not enough just to write numbers down. We want to write what we see down because that will help us understand and analyze our experiment later. And then we also have a spot for calculations. Calculations can be done in pencil, so I can take my pencil back up. So I'm going to do my RF for my red. Okay, and the way I'm going to write that is I'm going to write that RF, and I'm going to put a comma and write red next to it so that I know that that is for my red dye. And again, I'll do that 4.50 over my 8.11 millimeters. And I'll get my retention factor, and then I'll write the retention factor in the appropriate column back in ink. Okay, and then there's some analysis questions. Okay, for activity 2, for your food coloring, you'll notice that there is more than one line there for each color of the band. Okay, because maybe you might see more than one color in some of your Kool-Aid. And so for each band you see, right, for each color, use a separate line because you'll want to calculate the retention factor for each color that shows up for your Kool-Aid. And again, we have spots for observations and calculations. And then for activity 3, I have constructed a blank data table for you to fill out and set up any way you choose based on what you did in activities 1 and 2. Now, if this data table isn't large enough for what you choose to do for activity 3, you are free to construct your own. If this has too many rows or columns in it for what you choose to do for activity 3, that's fine. Just don't use the extra rows or columns. And then you'll do your experiment. And then there's going to be some analysis questions for you at the end. All right. Okay, when you are all done, you want to go ahead and clean up. Food coloring, Kool-Aid, table salt, you can go ahead and rinse that down your sink and then clean everything out. What you should do is clean everything with your tap water and then do a final rinse with your bottled water to make sure that all your glassware remains uncontaminated. And then when you're all done, those tips that you cut off, those can be thrown away. And, the extra pipette pieces can also be thrown away. And then don't forget, as you do your experiment, when you pull them out, again, you'll pull them out initially onto a paper towel, let them dry, and then you should have some tape and a piece of paper, because what you're going to do is then you're going to arrange your strips on your paper or on your piece of paper, and you'll end up uploading a couple of photos. So you'll do a photo for your food coloring control, you'll do a photo for your Kool-Aid, and then you'll have a photo for whatever you choose to do for activity 3. So you might need more than one sheet of paper depending on how you arrange your strips on your sheet of paper to take your picture. The whole reason we have you put them on the paper and tape them is because it's easier to see the colors if you are taking a picture on a nice white background. Because if you have a dark colored table or countertop you're working on, it's not as easy to see the colors sometimes. And so we're just having you use the paper to tape your strips to get a good photograph so that you can show the results that you received. Don't forget as well that there are 3 photos that you're going to need to take throughout your experiment. You're going to need to take a photo of your initial setup. You want to take a photo of yourself wearing your PPE to show that you are following proper safety protocols. And then some point in the middle of your experiment, you'll want to take a photo. And so maybe when you get stuff set up and it's in the test tube, you want to take a photo. Or maybe after you dissolve your Kool-Aid and you're spotting it on your chromatography paper, you want to take a photo. It's completely up to you what photo you want to take mid-experiment. Okay, all the experiments you have to take those 3 photos, and then it depends experiment to experiment what other photos you have to take. Like this one, again, you are going to have to take photos of your completed chromatograms and include those with your report. All right, so I think that concludes what I have to tell you. I'm going to go ahead and take off my gloves because I need to shut my computer down and it doesn't respond to my gloved finger. So that is our fundamentals of chromatography experiment. We'll be building on our chromatography next week when we do some stuff with spinach extract. So this is giving us the fundamentals to start understanding chromatography and what these retention factors are. And then we'll build on that with our next experiment using spinach extract. So as usual, if you have any questions, shoot me an email, shoot me a message on Canvas, stop by office hours, and I will do my best to help you out. So that is our fundamentals of chromatography experiment.